Good evening, everyone. Hi, this is Petko, live from the co-working space in Sofia Tech Park. This is the Clubhouse, a terrific facility. We are airing live our newest event. Today, we are focusing on a very special topic from the field of biotechnology. The subject is bacteriophages. Now, it took me a while to be able to pronounce this word, and it will probably take you a while to understand what bacteriophages actually are. But what they essentially are, let me try to explain that to you, are viruses that infect other microbes, so essentially a bacteria. So why are we tackling this topic, and why do we think that this is important? Most of you probably are aware that one of the most pressing public health concerns that is currently in the minds of scientists and medical staff across the globe is the growing threat of bacteria that is actually resistant to the existing treatments that we currently have, mostly antibiotics. So ironically, now we have to return to a technology that has been known prior to antibiotics. And these are exactly the bacteriophages, or these are the viruses that infect bacteria. This is one of the great promises now that we have in modern medicine, and we are hoping that this is one of the ways that we will be able to tackle this growing issue with new bacteria strains that are evolving to resist the use of antibiotics. This is a well-known issue. You are aware that antibiotics are very much overprescribed. They're available over the counter. They're also used in tremendous quantities in animal husbandry. So this has allowed for bacteria massively to be able to adapt. So new strains are coming out very often, and diseases that we have been able to treat very effectively are now coming back, and we are finding out that we are having a very hard time with dealing with them. So today's science is focused on bacteriophages as an alternative therapy of fighting these bacteria, and today we have a very special guest who is actually on the sort of on the edge of this well-forgotten but otherwise new science and therapy. We're speaking with Andrei Letarov. Andrei is a professor at the Lomonosov State University in Moscow, and he's also a head of laboratory at the Microbial Viruses Institute at the Vinogradsky Institute for Microbiology and Biotechnology in Moscow, Russia. Andrei, can you hear me?
Yes, I can hear you. Hello, everybody.
Добрый вечер, Andrei. As I said, this was a mouthful, so I think I didn't pronounce it pretty well, but I think I got that right? I mean, is that enough as a bio for you?
Yes, thank you. Let's probably pass to the bacteriophages that are
okay.
much more interesting than myself.
All right, you can go ahead and share your screen, and we can just go ahead and start.
Yes,
okay.
Just a second. Okay, I share my screen, and I launch my presentation. Will it work? Okay, it works. Can you see everything? Is it okay?
Yes, I can hear you well. I have a confirmation from the technical team that our audience can also hear you well. So you can go ahead and start.
Great, thank you. So bacteriophages. Bacteriophages, or the viruses of bacteria, and the word by the origin means the eaters of bacteria. It's not very exact description from contemporary view, but the term stood, and we call bacteriophages the viruses that infect bacteria. And from my own opinion, the bacteriophages are one of the most interesting and fascinating stories in biology. So bacteriophages, conceptually, are much larger than just therapy of any diseases or any other particular applications. And recently we passed 100 years anniversary of bacteriophage research that were initiated at the very beginning of last century. So by the end of 19th century, the microbiology was already a very well-developed field. It was not just an emergent science, but it was science that came directly to the hospitals. The routine practices of bacteriological analysis has been established in many leading hospital facilities, and the microbiological, biotechnological production of some vaccines, even sera and other products has been established, and even the viruses were already discovered by the very end of 19th century, but nobody understood actually what the viruses are. And in 1915 and 1917, independently or almost independently, these two researchers, Frederick William Tuort and Felix Derel, discovered the viruses of bacteria. And the research that was done on these viruses actually led to the establishment of the main concept of virology, the current understanding of the virus, speaking more precisely, the understanding of the virus as a transmissive genetic program that can travel from one cell to another. Unfortunately, I cannot stay alone on the history of the discovery of bacteriophages, because it's a really very very long and also fascinating story with a lot of controversies, discussions, even fights, travels, and so on. But I just will give a key point that the main idea of the discovery of the viruses and bacteriophages was the fact that researchers were able to identify some living viable agent that could pass through bacterial filters. Nowadays, bacterial filters are normally the membranes, but at that time they used porcelain candles. And these candles could be fabricated in such a way that the average size of the pore size was smaller than bacterial cells. And when the liquids were filtered through this kind of candles, mounted in amounts like that to fit it to the vacuum, for example, flask, so you could sterilize the liquid without heating them. And the researchers finally realized that in some natural objects, like in case of Derel, it was the suspension of feces of the person recovering from dysentery was filtered through this bacterial filter, and this apparently sterile filtrate was applied to the petri dish with nutrient medium alongside with a lot of dysentery bacteria. So bacteria grew as a continuous layer, so-called the lawn. And in this lawn, tiny holes that we now call plaques, and Derel called them in French taches vierges, did appear. And Derel immediately realized that he is dealing with a virus that infects bacteria. And his conviction is quite surprising, because he had no idea what the virus is. But he clearly understood that the thing he is dealing with is indeed the virus. So here, unfortunately, I should leave the historical field, but I strongly advise you to get more in, because it's really a very nice and fascinating story. And you can look an excellent artistic novel by Sinclair Lewis that was written in 1925, and next year, in 1926, Sinclair Lewis was awarded Nobel Prize in literature. And this novel tells us a story of a young emerging character, Martin Arrowsmith, a very young scientist who is working in the United States during the World War I, who co-discovers bacteriophages. And actually, those of you who are able to read in French can find the memory autobiography written by Felix Durrell that actually reads as an adventure novel, because Durrell was an adventurist, a traveler, and his life is full of very interesting turns and adventures. And a little bit more academical way, I presented the story of early research in bacteriophages myself in relatively short review that appeared in Russian journal Biochemistry that is available both in Russian and in English. Well, so now let us think about the bacteriophages from the modern perspective, what the bacteriophages are. And first of all, we can see bacteriophages in electron microscope. And here is a presentation of a paradigmal virus that you can find in most of biology textbooks, the bacteriophage T4. And the images of T4 and at different stages of the process of the penetration of T4 to the bacterial cell has been recorded using so-called cryo-electron tomography. So you can see that there is a cell, there is a phage particle. The cell is very small. It's artificially produced thing to facilitate this cryo-electron tomography. And the phage particle attaches to the cell surface and delivers its genetic material, in this case DNA, to the cell to provoke bacteriophage multiplication. And of course, T4 is just one of the examples of the phages. We can see much more of them in nature. And this image is kind of a visit card of my lab. This is an assortment of different morphological types of bacteriophages that were found in one single sample of the horse feces. And you can see that they are very different by size and appearance, but all of them follow the same outline of the structure. They have a head that is icosahedral structure, and they have a tail that can be long, flexible. It could be straight and contractile like in T4, and it could be straight and short, non-contractile. But anyway, the phages are built by the same principles. And this group of the phages that follow these principles are called, is called tailed phages, or the official name, the order Caudaviralis. The tailed phages comprise for about 95% of the all bacteriophages studied in the nature, and remaining 5% are shared between multiple families of non-tailed phages that are also of importance. But during our short lecture today, we will concentrate mostly on the tailed phages. So the bacteriophage, so the phage particle and so-called virion is kind of single action molecular machine. And the functions of these molecular machines are very simple. Firstly, phage should protect its valuable contents, the nucleic acid, the genetic material of the virus, from the harsh external environment. The second thing it should do, it should recognize correct cell that can support the multiplication of the phage. It's quite a difficult task because there are a lot of different particles in the environment. And the phage should choose the right one in which the genetic material can be effectively recognized and do its job. And the final step, once the cell is recognized, the phage particle should ensure the delivery of this genetic material through all the layers of bacterial cell wall. So conceptually, the functions of the phages are quite similar to the lunar landing module this here, that actually also the single-use machine that should deliver the astronauts to the moon. But in this case, it's probably better to take them back to the orbit, but it is the end. As it can be seen in the case of the lunar landing module, the phage particle is just a part of the complex phenomenon of the phage, because there are some spaceships, Earth control centers, and huge industry behind the lunar landing module. The same things are the infected cells and the complex biochemical processes that stand behind the simple phage particle. So let us look closely how the phage particles do work. The life cycle of the tailed phage, phages, takes place as follows. So there is a so-called lytic cycle in which the phage attaches to the bacterial cell, delivers the DNA to the cell, and this DNA is recognized by bacterial cell as its own DNA. It's just bacterium is also a kind of machine, and it is relatively stupid, so it cannot distinguish its own DNA from the phage DNA. It starts to execute a genetic program and registered on the phage nucleic acid molecule, and this program makes the bacterial cell to build inside itself new phage particles, and finally it determines the destruction, so-called lysis of cell, to liberate the phage particles to the environment, where they can find new prey, new bacterial cells to attack. I should note that all the production of tailed phages virions, so this lunar landing modules, can be achieved only in the lytic cycle. But some phages, in addition to the lytic cycle, can go through so-called lysogenic cycle, that is on the right. In this case, the phage delivers the DNA to the cell, and there is a point of choice. In some cells, it goes immediately to the Lytic cycle, and in some other cells, the phage interact with the own DNA of the cell chromosome, and integrates its genome into the cell chromosome, and the cell continues to live carrying the phage genome in its own genetic material. It may look like a time bomb, because at a given time point, in some of the cells that carry the phage genome, so-called prophage, the induction of the prophage may take place, and the phage cuts itself out of the chromosome to start the lytic multiplication. So in virology, we call the phages that can go only in virulent cycle. So they kill all their prey immediately. So these phages we call virulent. And the phages that can combine both lytic cycle and lysogenic cycle, we call temperate phages. This is a little bit more rigorous representation of the lytic and lysogenic cycle. And once again, I should tell that the virulent phages have only one wheel, only lytic cycle, and temperate phages invented the bicycle with two wheels, so they can have more sophisticated choices in their life. So the phages that are used in phage therapy are almost exclusively lytic or virulent. So we will speak about the virulent phages today, but we should keep in mind that the temperate phages that can integrate to the bacterial genome and to live together with bacterial for almost indefinite time also exist, and may exert significant influence over bacteria on our plant. So now let us look closer how the lytic cycle works. Here is a nice film how phage T4 attacks bacterial cell. This is a phage particle. It should attach to the cell and recognize specific molecule on the surface of the cell by so-called receptors by its legs or the fibers. And once the receptor recognized, the base plate opens and the tail contracts, pushing the central tube, the rod, into the surface of bacteria. So this tube reaches the cytoplasmic membrane of the bacterial cell, and the DNA is delivered from the head through the channel of the tail tube inside of bacterial cell. We can look at once more. What is the recognition of the receptors? The second set of the fibers deployed. Now the contraction of the tail follows, and the tube penetrates into the cell surface. To show you that it is actually indeed a machine, like very mechanistic structure, I can show you another film from one of the best structural papers in virology published so far. This is how the base plate, the bottom part of the bacteriophage T4 tail works. You can see that when it's open, there is a tiny needle at the end of the tail tube, and this needle actually is used to puncture the surface layers of bacteria and to make a way for the tail tube. Please continue the video. Actually, in this video, you can see how the base plate opens and closes back. It is made artificially to allow you to watch the process from different angles of viewing. In reality, the base plate is a single action device and only opens once when the phage attaches itself to the bacterial cell. And from this point, there are only two ways for the virus to successfully infect the cell or to die. It cannot close the base plate back and go seek for another bacterial cell. So it's no return point. And so let us imagine that infection is successful and the virus penetrated to the cytoplasm of bacterial cell. Actually, not the virus, but only the DNA molecule, because the bulk of the virion of this machine remains outside. And here we can see, please, see the process of the self-assembly of the bacterial virus inside of the cell. The whole assembly is a process orchestrated only by the interaction between protein molecules and protein and DNA. No control from the side of gene expression can be found here. So therefore, the assembly of the viral progeny goes ahead in a conveyor type. So some phages are already finished and ready to attack new cells, and some other phage particles are at initial stages of assembly. So you can see how the head is assembled, how the tail is assembled, and then the head should be filled with DNA. It is ATP-dependent reaction. The energy of ATP is used. Quite a lot of ATP is wasted for fill the head. And finally, the head is joined to the tail. to form the phage particle. And in case of T4, there is additional step of assembly of a complicated set of the fibers. Well, so let's go ahead. And once the phages are assembled, the cell blows up, it is lysed, and the liberation of the phage, also a controlled process is determined by the phage genes, but we cannot go in the details on the lysis mechanism. What is important that the cell gets completely distracted into very small pieces, and the phage particles can go ahead and to infect other cells. Now let us see how it works in nature. Recently, it has been discovered that in most natural habitats, the number of phage particles exceeds the number of bacteria by the factor of 2 to 10. And totally, there are about 10 to 31 phage particles in the biosphere. It is a huge number. So if we build a chain of all the phage particles put together in a single chain, the length of this chain will be about 200 million light years. Just to give you an example, the distance from our galaxy to the closest neighboring galaxy is just 2.5 million light years. So indeed, the number of the phages on the Earth is astronomical. And how these phages can coexist with bacterial cells that are still sensitive to them? Why don't the phage kill all the bacterial cells immediately? There is quite a simple answer. The phages are relatively tiny. Here is a structure of the phage G7C that was isolated in my lab and reconstructed in collaboration with Peter Lehmann lab at the time he was working in Swiss. And you can see that the receptor recognition devices, for example, this protein, are about only 20 nanometers long. So the pocket in which the surface molecule, the receptor of the cell can fit, is so tiny that the phage cannot see the whole bacteria. It just can interact with a particular molecule like a protein, and even not with the whole protein, but with a specific part, for example, protein loop or several sugar residues in polysaccharide molecules on bacterial surface. So the receptor recognition proteins of other phages are also very tiny. And this results in a phenomenon that the phages recognize not all the cells that are potentially suitable for their replication, but only those that carry correct specific receptors. And this leads to highly specific action of the phages. And in the environment, the concentration of the bacteriophages are relatively low if we speak about the absolute concentration of the phage that infect that very bacterial strain. And the concentration of the specific strain of bacteria also can be relatively low, because the total concentration of the phages and total concentration of bacteria are shared between multiple strains. So imagine if the phage, if the bacterial cell would be of the size of a human, so the phage will be the size of large orange, for example, that big. And 1 milliliter of the liquid will look like quite a large lake. So in a large lake, a human and orange can float for a very long time without any collision. And even if we increase the number of both sorts of the objects, the collisions, and in our case, a specific infection, will be just a little bit more frequent. So if we turn back to the bacteria, in real environment like an ocean, the probability to the bacterial cell to get infected over the time of the generation, so the time when bacteria divides, is quite low. So only a small number of the bacterial progeny is sacrificed to produce the phages. It is possible because the phage cannot accumulate in the ocean, because there are a lot of mechanisms of sink of the phages that can be inactivated by ultraviolet, that can die from inefficient interaction with bacterial cells or with other particles. They can be even eaten by the, some protozoans. As a result, the impact of the bacteriophage on bacterial mortality gets higher for more prolific, more ecologically successful strains of bacteria. And interestingly, as you could see a few slides ago, the phages infecting bacterial cell lyse the cell, and the biomass of the cell is chopped in a very small particles. This results by in the transition of the cell biomass to the pool of dissolved organic matter that makes a food for other bacteria. So the phages act as a kind of tiny Robin Hoods that rob the rich strains, ecologically successful strains, and take part of the goods, part of the organic matter from their populations to distribute this organic matter to less prolific species and strains of bacteria. Therefore, phage infection enhances the activity and diversity of the bacteria in the environment. And of course, there are a lot of interactions that are far beyond the simple killing of the bacteria by the phages. So bacteria, the phages coexist with bacteria for about 3 billion of years, and they elaborated a lot of approaches to turn this coexistence to mutual benefits. For example, the phage do a very important job trafficking the genes between bacteria in the process. This is called phage transduction. Basically, transduction is erroneous packaging into the phage capsid in the infected cells of a part of bacterial genetic material instead of the viral gene. And this genetic material can be delivered by this phage particle to a new cell, therefore enhancing the evolution of the bacterial genomes. The temperate phages, those that can integrate to bacterial genomes and live together with them for quite a long time, can also carry some specific genes that enhance the fitness of bacterial cells under certain circumstances. The process is called Lysogenic Conversion. And in addition to this, there are absolutely different and marvelous stories of collaboration between the phages and bacteria in nature. For example, the filamentous phage, point your attention here, the first mentioned during my lecture of non-tailed phages. Some phages are looking like pieces of the wire. So important pathogen, Pseudomonas Aeruginosa, frequently carries a prophage, in its genome carries a prophage of a filamentous phage PF4. And the particles of PF4 extruded from some fraction of Pseudomonas aeruginosa cells can participate in the building of the matrix of bacterial biofilm, a glue that joins bacterial cells together to facilitate the colonization of some surfaces. For example, the surfaces of the infected lungs in a diseased person. And in addition to participation in biofilm formation, this PF4 phage can mediate the interaction of the bacteria with immune system of the patient actually P4 phage particles penetrate, interact with macrophages and turn the immune response from antibacterial mode, when mostly tumor necrosis factor cytokine should be produced alongside with other signal molecules. And instead of this response, they trigger on antiviral response when interferons are produced. But antiviral response is much less effective to wipe out bacterial infections. So phage helps its host, its bacteria, to survive in a harsh environment of the human body, where immune system is aiming to eradicate the invading bacteria. Bacteriophages are associated with microbial systems in almost all the natural habitats, and the gut of mammal animals and people are not an example. Human viromes are believed to exert very multifaceted and poorly understood influence over the microbiome and even over the microorganisms, mediating the interactions of the virome with our body. And the emerging body of evidence suggests that in some diseases, like Crohn's disease, inflammatory bowel disease, the shift of the composition of the viral community can occur alongside with the shift of the composition of the microbiome. In our research, we also characterized the viromes of the feces of the horses, and we found that the horses are quite different from the humans from the point of view of bacteriophage ecology. So this highlights that the ecological rules of the games in different microbial systems, even in the guts of different mammal animals, that at first glance could appear quite similar one to another from the point of view of bacteria, but nevertheless, in these habitats, the ecology of bacteriophages are much different. Here is a little popular books about the horses. It was a by-side result of our research project. So to make a summary, I should say that bacteriophages from the modern point of view are not so much the enemies of bacteria, but rather they are tricksters that cheat, that betray, that may help, and they next moment it can change the side in a continuous fight of the life that's called evolutionary process. Fortunately, Felix Durrell in 1917 didn't know about all this complexity. And had he listened this presentation, he would probably never try to harness the phages to combat bacterial infections. But in fact, Durrell believed the phages are the part of the natural immunity and a real natural cause of recovery from bacterial infections. So he developed quite successfully the application of bacteriophages as a therapeutic agents. In his last monography that was, by the way, published in Russia, because he worked in the Soviet Union for twice in 1934 and 35, he tells us quite a nice story of success of his therapeutical efforts. Nevertheless, the technology turns to be not very reliable and was abundant on the West, specifically after the introduction of the antibiotic therapy that was very successful in solving the problem of bacterial infections. However, starting from the very end of 20th century, the emergence and the spread of multi-drug-resistant bacteria threatened to turn us back to the pre-antibiotic era. And phage therapy is reconsidered as an option to solve the problem, at least on temporal basis. The phage applications in medicine are considered simultaneously with application for food industry, for example, for decontaminating of the poultry products or preventing of the spoilage of fruits and vegetables. And also phages can be used in agriculture for multiple uses, like in veterinary medicine, and even to control the plant infections. But in any case, we should keep in mind that in contrast to quite a simple use of antibiotics, the application of bacteriophages every time is a kind of attempt at a crooked gambling in complicated play, ecological play between the bacteria and their viruses. And to be successful with this crooked gambling, we should really realize the rules of this game. Specifically in such a complex environment as a human or animal body, when not only the phages and bacteria are the players, but the immune system and the physiological processes intervene to alter the, for example, the concentration of the phages, the accessibility, the spread, and so on. The so-called active mode of the phage therapy, when very few bacteriophage particles delivered to the body of the patient could effectively multiply in situ to develop a huge viral population that will eradicate all the pathogen, is quite a rare case. It's hardly achievable in the real world. And in more realistic scenario, we should apply quite a lot of phages externally, because there are multiple mechanisms that limit phage multiplication in situ. And to get clinical success, we should add more and more phages artificially. For example, in the biofilm, phage can attack some cells and multiply and successfully kill neighboring cells. But then when they reach, for example, edges of some cloud of the biofilm, the phage can get dispersed, and to continue the destruction of the biofilm and the eradication of the pathogen, we should wait for new phage particles to come, for example, from the blood. The dosage and working concentration of the phages and pharmacokinetics are very important factors that complicates the wide use of phage therapy. So the main advantage of the phage therapy compared to antibiotics is the fact that in the phage therapy, we may counterimpose phage evolution that is very powerful to the evolution of the bacteria. So the bacteria can easily adapt to get resistant to the phage. This adaptation is much simpler than the adaptation to the antibiotics. But bacteria cannot evolve to get absolutely resistant to all the phages. And we normally can find more and more phage to kill the evolving bug. But at the same time, as I told you, the phages naturally didn't evolve in such a way to be effective killer of the bacterial population. In nature, they are not seeking to eradicate their hosts. They are more keen to coexist with them. And this makes a successful phage therapy quite complicated and scientifically, rich topic. So there are a lot of issues and a lot of research needed. And I will give you several simple examples of the phage therapy. The classical target of phage therapy may be, for example, surgical infections. This image is a kind gift of the Polish team of phage therapy from Wrocław, and they show the treatment of the long-lasting antibiotic-resistant infection in a patient, and you can see that a very long course of phage therapy eventually led to clinical resolution of the situation. But in reality, we can not only use the phage in therapy as a killing agents like antibiotic, but we can also play with bacterial adaptation to the phage pressure. And we may take a benefit from the fact that the bacteria that try to avoid the killing by the phage open another weak points to synergistic attack. For example, some phages may target, may use as the receptors some surface bacterial proteins that are involved in bacterial virulence or in antibiotic resistance. The first well-known example of the last situation was this very nice life story. This guy, whose name was Thomas Pettersson, he's a professor of some humanitarian science, and one day he got a very nasty disease, kind of pancreatitis during his vacations. And as a complication, he developed a peritonitis caused by multidrug-resistant Acinetobacter baumannii. And fortunately, his wife is an epidemiologist, and she was able to arrange an experimental treatment of his husband that was performed by a large team of scientists and doctors in Texas. And eventually, after several weeks of the treatment, when the researchers several times changed the bacteriophage mixture composition to follow the adaptation of this bug, the bacteria was driven to the sensitivity to one of the antibiotics to which it was resistant at the beginning. So the doctors added reintroduced the antibiotic to the treatment regimen, and this combined action of the phage and antibiotic led to a cure of the patient. The same trick was recently published to treat the infection of some aortic graft colonized by Pseudomonas aeruginosa. In this case, the phage-induced antibiotic sensitivity was a deliberate action. So the researchers deliberately found the phage that was able to provoke return of the bacterial strain to antibiotic resistance and antibiotic sensitivity. So, and this indeed worked very nicely. So where can we go further? Everything that I described before were the attempts to use natural diversity of the bacteriophages. And in fact, natural evolution of the phages is a very powerful thing. But in such a complex environment as a human body, the other features of the phages, like for example, the visibility to immune systems that can eliminate the phages quite effectively in some cases, are also of importance. And it may be a beneficial strategy to find several good phages and engineer them to employ them against different bacteria, different bags. Actually, the first example of the usage of the engineered phage to save a patient with so-called cystic fibrosis is an inherent lung disease that normally is complicated by bacterial pneumonia of different etiology. And in this particular case, a young girl, 15 years old girl, was infected with Mycobacterium abscessus, and it was impossible to find a good virulent phages to control the infections. So the researchers designed virulent mutants of temperate phages by editing the bacteriophage genome and managed to help the patient. But the designing of the lifestyle is not only the one option, and the most attractive possibility is the designing of the host range. So we can alter the proteins of the phage that recognize the surface receptor of bacteria. In such a way, we can use a one platform phage to retarget it to different bacterial species, or at least to different bacterial strains, and to come up with a second generation of the phage therapy that will be based on more characterized, on more adapted bacteriophages to reach the clinical improvements. So, the very last words. So, where are we now? In some countries, for example, in Russia, in Georgia, the phage therapy never ceased. And even nowadays, the phage-based drugs are available in the pharmacies in Russia. I used to buy them sometimes in Moscow. And most of these drugs are produced by a company called Microgen. But these drugs that are currently commercialized are somewhat old-fashioned. They represent very empirical phage therapy. However, recently, Microgen decided to develop their production, and they established so-called Biological Resource Center. It will be a research unit in Ufa city in Russia, where they're aiming to add detailed genetic characterization of the whole collection of the bacteriophages, centralized management of these phage resources, and they're aiming to improve the bacteriophage cocktails to operate with the phages that are exactly known and delivered in correct scientifically chosen concentration and so on. And we hope that this will at least partially reveal the problem of the phage resistance that even now is very, very important in Russia. For example, during the COVID outbreak, most of the people dying at artificial lung ventilation dies not from the viral effects, but from the effects of the uncontrollable bacterial pneumonia because antibiotics just stopped to work. So this is a very last point of my scientific talk, and I may give you some reference of the books that you may find if you would like to know more about the bacteriophages. I see that I failed to fit my talk in 40 minutes, but now it's over. And thank you very much for your attention. And I'm ready to answer your questions if you have some.
All right, thank you very much, Professor Letarov. Yes, you didn't match the 40-minute mark, but I think it was a very interesting and informative 1 hour. So thank you for that. Before we move to the questions, just a quick side note and a quick thank you to Microgen and to Dr. Simeon Baburov from Substancy Pharma, who essentially helped us so we can get in touch together so we can give this talk to the people. So a quick thank you to them. Now, before we move, Professor, to the more scientific questions, I want to review something that is of particular interest to me. This phage therapy has been known, as you already showed in the presentation, for quite a while now since the 20s. But it was mostly within Eastern Europe and the Soviet Union. And most of the studies that have been done and have been published on phages have been published in Russia, and this partially explains why this therapy hasn't been, well known to the so-called Western medicine for a while. Now, can you explain why is that as a historical phenomenon, so to say?
What kind of phenomenon? The therapy was initially invented in the West.
Yes.
So Durrell is a Canadian scientist who worked in France mainly.
Right.
And he was very enthusiastic about the phage therapy. And many companies in the 1920s and 30s started to produce phage drugs. One of the companies was controlled by Durrell himself, but he claims that many producers didn't follow the rules he invented to choose correct phages and to produce them. And even if they did, at this time, the understanding of the phage biology and the complexity of the interaction with the phage with bacteria and with the human body were strongly inferior compared to contemporary understanding. And even nowadays, we do not understand these things enough to make this therapy easy to use.
Right.
So, and when the antibiotics arrived, it was a miracle because they are much simpler for doctors. And the simplicity means everything in medicine, because the doctor can really bring help to the patient without complicated discussions about science, laboratory testings, and long time to find correct phage preparation. Why didn't the phage therapy vanish in Eastern Europe? I don't know exactly.
If you allow me, Andrei, one of the things that I actually read when I was doing my research was that this therapy was mostly used in the Soviet Union and in Eastern Europe because antibiotics were not widely available at the time in these areas, and therefore the focus was on phages.
This kind of misconception, because the phages were indeed intensively, the phage therapy was developed intensively in the 1940s and the end of 1930s and 1940s, during the wartime, for example, when antibiotics were indeed poorly available, but they were poorly available in the world. And starting from late 50s, when phage therapy still existed somehow in the West, the last production of phage drugs stopped ceasing in France in 1970s. But nevertheless, the antibiotics were quite abundant in 60s in Soviet Union, and Soviet government did extensive effort to establish their own production of antibiotics, but because it was strategically important resource for the case of potential war. But nevertheless, the phage therapy were not completely abandoned. Fortunately, and probably in a part of this came from some kind of inertia of Soviet planning system, and on the second hand, from the understanding that phages sometimes were better from the point of view of safety, and in some rare cases, even from the point of view of efficacy. Some kind of infections were quite difficult to clean with antibiotics, even if the bacteria were sensitive. But the phages could penetrate and multiply inside. But this were quite rare cases, but they nevertheless existed.
Yes,
The,
and we can see these challenges now. So if we have to now start speaking about the challenges that this phage therapy is facing and why it's not yet widely available, there are a few aspects here that I would like to touch on. The first one is sort of the logistical challenge that we have to face here. So phage therapy, if I understood correctly, is a form of personalized medicine. So you have to find the correct phages to fight the specific bacterial infection that a patient has, right? Did I understand that correctly?
yes and no. Actually, the phage therapy in its current form in Russia is not personalized, individualized therapy.
Yes.
You can just buy phage mixture in a drugstore that is designed in such a way to cover potential repertory of, for example, Pseudomonas Aeruginosa strains or pyogenic bacterial strains. This is not the most effective strategy.
Okay.
And the strategy with individualized therapy seems to be more promising.
Yeah.
So even in Russia, and including the Microgen company, despite the fact that they mass produce these general drugs, but they also recognize the need for individualized phage therapy. But individualized phage therapy is a labor-intensive and I would say quite expensive therapy, because you need the effort of very qualified staff.
Yeah.
And it is very hard from the legal point of view, because you know you need to establish the regulations that would allow routine use not of some very defined drug, but just a technology that includes continuous finding of new phages in the nature, or probably in future new engineering. And if you don't do so, you lose the main point of the therapy, when you can counterimpose evolution to the evolution of the bacteria that are seeking for resistance, the evolution of the phages that can find new ways to penetrate to the cell.
Yes. Yeah. So essentially, you are saying that it's a logistical challenge because here essentially we need to first have an enormous amount of samples and a huge specified database of specific phages and specific bacteria, so we can match them together. So this sounds to me like a bioinformatics issue as well, and this is why it's difficult to make it personalized and to scale it up.
It would be an ideal solution to shift it to the bioinformatic field, so to do it in silico. But nowadays, we need to have physical collections of the phages, and to experimentally test the sensitivity of the strains. And in the ideal world, for example, like Polish team in Wrocław is trying to do, they say, under the umbrella of Helsinki Declaration that allows to use experimental treatment in the cases if there are no other options. So what they're doing, they treat people with chronic and resistant infections where other options just don't work. But in any way, it includes bacteriological investigation and analysis of this specific strain, choosing phages against it, preparing the mixture, and in complicated situations like with this professor about which I just told a couple of minutes ago, with Acinetobacter baumannii peritonitis. The researchers closely followed the treatment, and they several times found new phages to tackle the in situ resistance. So in simpler cases like surface infections or some not very complicated pneumonia, for example, in situ resistance can be not a major problem. And phage therapy works much better in the circumstances when you do not need to eradicate with the phage the bacterial population completely. It's kind of non-natural behavior to the phage to kill the bacteria completely. But if you, for example, disintegrate biofilms, if you reduce the density of bacteria, you reduce local intoxication, and you allow immune system factors like macrophages and so on to work efficiently. And maybe if you bring the bacteria under the sensitivity to normal drugs by these combined approaches, you can act much more effectively than with the phages alone.
Yes. Okay. So one of the other challenges that I read about when it comes to the phage theory is that our current understanding of the coevolution between bacteria and phages is fairly limited. That we don't understand as precisely as we want to how they actually co-evolve and what are the mechanisms of coevolution. Can you address this this criticism somehow? I mean, are we that far behind in understanding how they co-evolve?
From a theoretical point of view, we have quite a nice picture of the mechanisms of phage evolution. So absolutely not complete, but nothing in biology is complete. But when we are speaking about the phage therapy, we need the detail matter. So we need, for example, predict how fast the resistance will develop. And if it does, will the resistant bacteria be more nasty or they be weaker than original strain? And the good news in most of the cases, the resistance that's selected in situ immediate resistant strains are weaker, they're less virulent, they're more sensitive to antibiotics than original strain. So this also helps. But there are some exceptions, unfortunately.
Yes, yes. As always, there are some exceptions. Let's talk about our immune system. Now, our immune system is also known to react and to form immunity to certain types of phages. Can this actually reduce the benefits of a phage therapy?
Unfortunately, yes, this can. There are some controversy, some discussions, because some scientists demonstrated that in some particular systems, the outcome of phage therapy was not so much compromised by pre-existing antibodies, but in some other circumstances, and this depends on exact location and of the infection, for example. And details of the patient. But generally, it is better to choose the phages to which the antibodies are not present, at least at the beginning of the therapy. And in many cases, the therapy regimens are not so long as I showed on my slides, or not the weeks, probably a few days. The period is shorter than the time necessary for development of the antibody response. And just yesterday, I was speaking to a colleague that is involved in experimental individualized therapy in Russia. And he told me that he prefers always check if there are antibodies for the phage he is going to apply. And if they are present, he's trying to find another phage strain.
So do you think that this should be part of the standard way that we approach the therapy, that we test for antibodies first?
It depends on therapy of what.
Right.
If
okay. Yeah.
But
okay.
In some cases, yes, of course.
Okay. Now that we know, I think you touched a little bit on this in your presentations, but we now know that phages can be a vehicle for horizontal exchange of genetic material, essentially. So, can they be part of? Can they actually play a role in the diffusion of antibiotic resistance in bacteria?
It is a very interesting question. In theory, they can, and you can in the lab you can easily transduce antibiotic resistance genes as well as any other genes, and also you can easily artificially design a temperate phage, the phage that can integrate to bacterial genome in such a way that this temperate phage will carry the antibiotic resistant gene. Actually, we even use this as a nice research tool. So, it's very convenient when you form your lysogenic bacteria. You can distinguish them because they are antibiotic resistant now. But in the real world, very few, if any, antibiotic resistant genes were found in the phage genomes. So they are almost never the part of the so-called lysogenic conversion, about which I was speaking. Nobody knows why.
Yeah.
But on the other hand, the transduction probably can effectively work in the field also. So you can transport the gene from one bacteria to another using the phage transduction. And therefore, it is quite important, and it would be probably a good practice when you choose the phage for therapy purposes. You should probably choose the phage that are not able to so-called generalized transduction. So in which the mechanisms of the DNA packaging are precise enough, or there are some other mechanisms that prevent erroneous packaging of bacterial DNA and gene trafficking. In order to not to get the situation worse because of your therapeutical attempts.
Yes. So is that a easy thing to do? I mean, going back to the scaling up question, if we have to analyze if this is potentially possible to happen with this specific phage, is that a complex test that has to be performed?
No, it's not a complex test at all, but it's not a complex in theory or the demonstration of principle. And when you need to really do it in massive amount, it can be
difficult.
Kind of a challenge, because it's not so easy to ideally to make a standard to make, for example, technician to do it for you on a large scale. You need to understand what you are doing in each case. And this is the benefit of usage of engineered phage, because they are once checked carefully, and so you are sure that they are safe.
They're specifically designed. So this exchange of genetic information, you're saying essentially that it is theoretically possible, but it's not really observable in the life world a lot?
Once again, the gene traffic is possible and observable.
Okay.
But the carriage of antibiotic-resistant genes in temperate phages in terms of so-called conversion that could be expected, because temperate phages frequently include different genes that help the lysogenic bacteria to survive. For example, many bacterial toxins are actually encoded by the phages, like diphtheria toxin, cholera toxin, and many others.
Right.
But there are a lot of phages that encode toxins, but very few, or even none, encode antibiotic resistance. We don't know why, because artificially you can easily force them to do this.
Right. Okay, you did mention that phages can actually help some bacteria. Can you elaborate a little bit more on that? I mean, how can they help them, and how is this beneficial to us?
How they can help them? Well, I gave you just one example of the participation of the phage in the development of the bacterial biofilm. The biofilm sometimes called a city of bacteria. So what happens, the bacteria, when they colonize some surface, they attach and they change their physiology to build a kind of colony. The cells are surrounded by complicated things, so-called bacterial matrix, biofilm matrix, something like glue made of polysaccharides, even DNA and other molecules. And this PF4 is not only example of the phage that actively participates in biofilm matrix. So there are other phages that can be actually structural components of the matrix. But moreover, some phages give bacteria kind of a signal. So when bacteria face the phage attack, they are trying to enhance biofilm making, probably to avoid too high phage-induced mortality. But there are some ideas that this could be actually not so much the adaptation of bacteria, but adaptation of the phage, because the phage is not interested in killing the bacteria immediately, but it would like to remain longer in the site where the concentration of the bacterial cells is half enough to support this continuous multiplication. So it's kind of domestication of bacteria by the phage. It doesn't act like a predator, but it acts like a smarter species that domesticates something like cows to keep them and eat them gradually.
Right. Okay, you did show us some medicines that are currently being sold over the counter in Russia. Not over the counter, probably with a prescription. I'm sorry, I guess I missed that point there. But can you briefly tell us or remind us what are the most common conditions or diseases that these are being prescribed for?
Okay, first of all, I never was asked for prescription in a drugstore, so they do not care about the phages. They can ask about the prescription for antibiotics, and this is logical.
Right.
The phages they are concerned.
For phages, they don't require prescription?
Not at all. So
okay.
I never was requested to show the prescription for phages. So the most obvious applications of the current phage drugs are to the infections localized somewhere topically. So it could be some small wound infections that where phage can be applied as an ointment or you can rinse the small wound with the phage. You can apply them to treat intestinal infections. In this case, you should drink the phages. They are relatively safe. Have very few, if any, side effect. The more controversial is the treatment of, for example, urological or pneumonia, because currently we don't have any phage formulation for injections. So to do so, you need to develop highly purified phage preparations. Otherwise, the endotoxin, so lipopolysaccharides, for example, present in the crude phage lysates may provoke toxic shock and other complications. So currently, we don't have on the sale any injectable phage preparations. Nevertheless, if you look to the sheet of the supplied with the phage drugs, they suggest to drink it in the hope that the phage will be transported by blood from intestine to the sites of the infection. Maybe it may occur in some specific cases. For example, in some patients in critical stage, probably the penetrability of the gut is enhanced. And in some inflammatory gut disease, inflammatory diseases is also can be observed. But in more or less healthy people, the transportation of the phages from the gut to blood possibly indeed occurs. But the strength of this process is not high enough to ensure a reasonable concentration of the phages in the blood. So I have a significant doubt in the treatment of the phage, of the I would say of the infections localized outside of the of the gastrointestinal tract, or not on the surface where we can apply the phage topically by drinking them.
So it has to either be in the intestines or on the surface of the body, speaking of the current stage of development of phage drugs.
Exactly. For example, Staphylococcus phages are quite good. Just a peculiarity of the Staphylococcus physiology, they have some quite conserved molecules exposed on the surface. So there are phages that cover maybe 95% of the strain.
Right.
Very broad host range strain, if we consider just Staphylococcus aureus. And this can be used, for example, in the case of diabetic foot syndrome, in the case of skin staphylococcal infections, in some pharynx staphylococcal infections, and so on.
Right. And because we have a question here from the audience that is currently watching us, you did mention that there are very few side effects that are known at this point. But just as an additional question to this one, why are we not seeing a lot of large scale research and clinical research being conducted right now in Europe across the world? So meaning that if we know that there are very few side effects, I mean, how do we know that when we essentially have limited research being done on phages?
I wouldn't say that the research is so much limited. There are a lot of quite powerful scientific research groups in Europe, for example, in University of Leuven by Rob Levin. There is a group in Pasteur Institute by Laurent Debarbieu. And there is a very nice group in Portuguese led by Joana Azareda, who developed different therapeutical approaches. But what about the large trials? It's a very complicated stuff, and it needs a vast investment and probably a strong political will. Because first of all, I spoke a lot about the complications of the phage therapy, the complexity of phage therapy. So it's not very easy to get it effective. But even if you come up with some design, there's immense regulatory problems. So it's very difficult to get authorization to perform such trial. And if you get it, you, for example, need to produce your phages under the GMP standards, good manufacturing practice. This is quite expensive and difficult, and not actually really needed in all the cases of phage therapy, but nowadays it's requested. So a few teams are able to get through all these barriers too.
Now, do you think that such regulatory change and more focus and money is due now, considering the issues that we are having with antibiotic-resistant bacteria and all of that? I mean, do you think it's time now to actually start to heavily invest in this type of research? I'm asking about your personal opinion here.
Yes, I see. My personal opinion is that yes, because I realize that the phage therapy is far from being a perfect medical technology. But I don't see any real alternative that can solve the problem in terms of next couple of decades. And even now, we can see very harsh consequences of the antibiotic resistance crisis. For example, I heard just during my professional conversation just yesterday that one doctor, the head of some intensive care unit involved in COVID treatment, just told to somebody that he refuses to administrate antibiotics to artificially ventilated patients just because no antibiotic work, just useless. And they lose a lot of lives that could be saved had COVID strike, for example, 20 years ago.
Right.
It is quite strangely, but I'm not speaking about the total number of casualties, but people who are at artificial ventilation would have higher survival rates 25 or 30 years ago when antibiotics still worked. And this moves to less severe conditions because there are we really approaching to the state when somebody relatively healthy can die in a few weeks because of the complications of minor surgical interventions, like, for example, appendicitis treatment.
Right.
And the phage therapy, despite all the weak points, can at least partially help right now. Even with approaches of Durrell, almost 100 years old, we can save a lot of lives. But later, I really hope that some more handy approaches, maybe, for example, rational drug design, will yield a new generation of antibacterial chemotherapy, and probably phage therapy will remain like some specific product for example, for pregnant women.
For specific cases.
For specific cases,
Just a step back. Because I didn't hear you correctly about this new hope. Did you say racial
drug design? So
what do you mean by that?
We can use bioinformatical and computer modeling to invent in silico the molecule that could probably specifically inhibit some specific enzyme in this bacteria, and to synthesize it, to test it rapidly, and to come up with some antibacterials that are not derived from nature but completely designed by humans. Right. And there is a concept, for example, of narrow spectrum antibiotics that will spare our natural microbiome from side effects of conventional antibiotic treatments. So there are many ideas, but unfortunately, I don't know about any single antibacterial drug that was brought to the market by these technologies. It probably will require some decades more to solve the crisis. And
so we are very far away from that. Is that what you're saying?
Maybe 20, 30 years, maybe close. Nobody can predict, but we still need to survive this 20 years. And the phages might help.
True, yeah. Can you, let's just take a step back. Now, we are seeing that when it comes to the pipeline of new antibiotics that are being developed right now, the pipeline is essentially dry. I mean, it's very few new products coming out. Why is that? Why is it so difficult to develop new antibiotics, I guess is what I'm asking.
It is difficult and it is expensive because you first of all need to find one. Yes, there are some more high throughput approaches compared to old Waxman procedure. Some projects using, for example, microfluidic devices that allows to broader screen for potential and antibacterial substances in nature produced by, for example, by other bacteria.
I'm sorry. Let me just interrupt you here. So you're saying that the only we are now capable of developing new antibiotics only from, so to say, natural sources. I mean, we have to find them. We can't really make them in a lab.
Almost all, or probably all, the new classes of antibiotics were found in the nature. In the lab, you can modify them to for example, to develop new generations of, for example, beta-lactam antibiotics starting from penicillin were found initially in nature, but then chemical modifications were elaborated to make them more active and to avoid some mechanisms of resistance.
Right.
So modern beta-lactams are resistant to activity of beta-lactamases of many bacteria, but bacteria develop more sophisticated beta-lactamases more fast than we modify our beta-lactam antibiotics.
Yeah.
And new classes of antibiotics were not introduced into the practice probably over 15 years or something. So we are living with modifications of old ideas, so old principles and antibiotics.
Now, is that due to the fact that we're not looking for them, or is it a matter of loss of biodiversity even? I mean, what are we talking about?
Probably we exhausted a repertoire of the molecules that were easy to find, the first.
Right.
The second reason, most of the molecules you find are not suitable as drugs. For example, they are toxic.
Yeah.
And then if you find a good one, you need to make research, to make a clinical test, to develop the production. And from the point of view of the pharmaceutical companies that are not very economically effective, because people can, for example, use new generation antibiotics probably a couple of times of the life.
Mm-hmm.
The consumption of these new antibiotics not so important compared, for example, to cardiac therapy that people take on a daily basis for the lifetime.
Yeah.
So they have a tendency to invest to more reliable and more cost-effective products. So the second half of the problem. And eventually, now we come back to the usage of some antibiotics that were discovered ages ago, for example, like colistin. The colistin was abandoned because it's very toxic. But in some cases, doctors have to use it again and again, because despite, for example, the acute renal toxicity, because there are just no other options remaining.
Yeah.
Somebody is infected with some bug that only colistin sensitive.
That doesn't sound very good. Now, when it comes to the future promises of biotechnology, I mean, can we? You are saying that right now we are capable of modifying existing molecules that we find in nature, but on a conceptual level, at least theoretically, can we develop artificial antibiotics?
We started with this, probably yes, but not right now. But let's probably get back to the phages, because I'm not really a specialist in rational drug design.
Sure,
and
sure. We have plenty of questions here, and most of them are related actually to the alternative uses of phages that you briefly touched upon, like agriculture, food, and etc. So, can you give more specific examples on these novel applications? We have a specific question here about the food industry, for example. How can they help?
Okay, for example, if you own a poultry plant, so you produce hundreds of tons of chicken meat per day. And one day, a sanitary inspector finds some salmonella on your products, and you have to
kill them all.
To remove, throw them out, and to stop your facility to clean it from salmonella. It causes immense economical cost. But you may try to spray salmonella phage to this meat, not to administrate them to the chicken, but just treat the meat, and to eliminate salmonella or some other specific pathogens, aiming at such bacterial species that are highly controlled by.
Right. So you essentially saying we spray the facility, like.
You may spray the facility, or you may treat your production, the meat.
Okay.
You can, for example, submerge them to the bath with the phage or spray with the phage, and this really can work. There were some other developments that are not yet really applied practically, but you can even extend the shelf life if you develop the phage therapy that would target the bacteria that causes spoilage. Of course, you cannot target all the bacteria that spoil the meat because they are very numerous.
Yeah.
The tasty substrate that will be consumed anyway. But you may target the groups that appear first. And the time from the beginning of the storage point when you reach there is a parameter that means the total number of bacteria that can be plated from your meat with tongue twisting abbreviation. So this total bacterial contamination should not exceed some limit. So if you shift by day or two the moment when you reach this limit, you save a lot of meat from the waste actually, and avoid a lot of economical loss.
Well, that sounds actually great, because I mean, concerning the food industry, that seems to be a big issue, right now. The risk of contamination. Considering how large scale is the meat production. So
I can see that there is also a dark side of the medal, because the phages are the major problem in dairy industry. Because in dairy industry, the phages may cause a failure of fermentation. For example, when you make something like yogurt or other products that cannot use, where you cannot use the sterilization of the milk.
Yes, it will kill the bacteria, right?
It will kill the milk. So bacteria can be added later, but in some technologies you cannot boil the milk.
Right, yeah.
And the raw milk may contain 10 to 4 phage particles, specific, for example, for lactococcal cultures that are used for the fermentations.
Right.
And if the phage concentration reaches some limit, about, for example, 10 to 7 to 10 to 8, when they can stop the growth of the bacteria, they cause a failure, and in place of having a good yogurt, you have some disgusting semi-fermented product. Right. And in this case, you need to stop the facility, to clean it out, to change the starter cultures. So there's a complicated industry around rotation of the starter cultures to evolve phage lysis in dairy industry.
Yeah, so we don't use it for milk, but we have multiple other applications. Professor Letarov, we are unfortunately out of time now, but I want to thank you for this very interesting, at least for me, conversation, and for the talk you gave us. You certainly presented a topic that I wasn't familiar with at this point, and I believe that our general audience is also hearing something new here. So thank you very much for this talk and for your participation. It was a privilege.
Thank you very much for the invitation. It was a pleasure of mine to meet you this evening.
Very good, very good. Well, hopefully we will be able to meet in the future again. I'm going to ask you to stick around. I just have a few more things to say to our audience, and then we can chit chat and say goodbye in a proper manner. But thank you. Okay, ladies and gentlemen, as you know, as per tradition, now I'm going to give you a quick overview of what's coming next. So I'm going to ask the technical team to show us the next visuals created by our awesome designers. So let's see what's happening. Well, of course, first we have to thank the people that are making this production possible. So thanks to the National Culture Fund of Bulgaria. They are being very generous to our organization and are helping us so to say, run the lights here. So thanks very much to the Nature Culture Fund, and of course, thanks very much to the Sofia Tech Park here. We are filming in the clubhouse, and we are using the grow working space as well. Awesome facilities, and we want to thank them for accommodating us and for being so accommodating to us in general. So thanks to Sofia Tech Park. Our next event is on the 16th of February. Again, we are starting the live stream at 8 o'clock, tuning in to the Radio Universe. We're going to be speaking with Professor Gallagher, who is from, I believe, University of Dublin. Correct me if I'm wrong, Nicola. Yes, that is correct. So join us at 8 o'clock. We will be listening to the universe. I mean, look at this design. It's insane and psychedelic. Okay, if you are a podcast person, you can listen to our podcast. We have currently 3 major formats. We have a culture podcast, and for speaking about cultural intellectualism and all the rest, we have the Vox Nihili format, where we discuss the interaction between science, ethics, and philosophy. And we, of course, have our weekly science podcast with Nikola Kerekov. So if you want to hear the last interesting news in the world of science, you can catch us on our podcast. Thanks to our patrons, we have tried to do something additional for the people who are supporting us financially on Patreon. We are now doing an extra podcast episode together with Nicola, especially for you guys who are helping us out with running the lights here, essentially. So check out for our patrons our special episode, Recycling of Space. I think it was an interesting conversation with Nicola. Thanks again, guys. So you can see your names here. Hopefully, there will be more names coming in. Thanks to all the people that are supporting us, and I hope I will see you next time as well. Before that, of course, follow us. In every social media that you use. Check out our website to see the next event. And I think that was it from me. Stay healthy, and I will see you next time. Thank you. Bye bye.